Hello everyone, myself Dr. Pradeep Kumar MR, working as Assistant Professor in the Department of Pharmaceutical Chemistry at Kelly College of Pharmacy, Hubli. In this, I will be discussing with respect to qualitative tests carried out for identifying the carbohydrates. Before I could start, I just want to give the definition and the classification of the carbohydrates. Then I will continue with the different qualitative tests carried out for identifying the carbohydrates. Let us see the definition of the carbohydrates. Carbohydrates are the polyhydroxyaldehydes or ketones or the compounds obtained by the hydrolysis of the same. These carbohydrates can be classified into monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Polysaccharides are further classified into homopolysaccharides and heteropolysaccharides. So, having got this basic information with respect to the carbohydrates, we will look into the practical aspect of identifying the sample of the carbohydrate provided for carrying out the qualitative analysis. So here I will be discussing with you the qualitative tests which are carried out in the laboratory to identify the given sample of the carbohydrate. So in this session I will be discussing the different qualitative tests. In that I will be stressing upon the principle involved in each test, followed by the composition of the different reagents which are used in the particular test, then the procedure or the test procedure what we are following to carry out that particular test we are going to see. Along with that we will be seeing the type of the observation and the inference what is there with respect to that particular test. Also to support that I will be showing you the images of the color formed or the precipitate formed in that particular test through the image which can make you to feel that. Let us see the first test. The first test is Maulish Test. This test is a group test for carbohydrates, and this is given by all the carbohydrates whether free or bound to such substances as proteins like glyco or mucoproteins and lipids, glycolipids. So in this the principle involved, you can see the reaction here. The polysaccharides and disaccharides are hydrolyzed by concentrated sulfuric acid into monosaccharides. The monosaccharides are dehydrated by the acid to form perfurol or its derivatives, which later condenses with alpha naphthol to form a violet colored complex which is appearing in the form of a ring at the junction of two liquids. So actually what the reaction is occurring, what is the principle behind that I just gave you. Here I am showing the same in the form of a chemical reaction. So as already we know that carbohydrates are either containing the aldehyde as the functional group or ketone as the functional group. When they are treated with a concentrated sulfuric acid, there occurs a dehydration which results in the formation of the perfurol derivative, which on further treatment with the alpha naphthol gives rise to a violet colored complex which actually appears as a violet ring at the junction of two liquids. So here we need to know little bit regarding the composition of the reagent what we are using in this particular test. So here we are using the Maulish reagent. So what is this Maulish reagent? How it is prepared? It is prepared by dissolving 1 gram of alpha naphthol in 95% ethyl alcohol, and then the volume is made up to 100 ml, whereby the Maulish reagent is ready for the experiment. Next, let us see the procedure. What is the procedure involved in this Maulish test? Means how we can carry out in the lab. So let us see the test column first. Here we are taking first a clean test tube. To that we are adding 2 ml of test solution. That is, whatever the compound is given, its solution we are preparing to identify that. That is what we are taking as a 2 ml of the test solution as, plus 2 drops of Maulish reagent. Just now what I discussed with you with respect to its preparation, the prepared Maulish reagent is added, that is only 2 drops, plus 1 ml of concentrated sulfuric is added. The care should be taken that the addition of this concentrated sulfuric acid should be along the side of the test tube, not directly. So when we add these things in the sequential order as explained, we can observe a violet ring which appears at the junction of two liquids. When there is a formation of the violet ring at the junction of two liquids, the inference we can get is, it is an indication that the carbohydrate is present. Here at the top I have shown, so when you carry out the test in the test tube, so at the end you can observe this type where you can clearly see there is a violet ring which is seen at the junction of two liquids. So which in turn confirms the presence of carbohydrate in that particular sample. Means we know that we are carrying out the qualitative test for carbohydrate, but first we should see that the given sample is carbohydrate or not. So for that reason we will be carrying out this Maulish test, which is given by all the carbohydrate. First we need to confirm that the given sample is carbohydrate. For that reason this test is carried out. Next test, that is solubility test. So coming to the solubility test, this is a simple test where we are taking the sample, that is a solid sample, whatever the sample is given as a carbohydrate for the identification test. To that we will be adding the 2 ml of water. So after the addition of the 2 ml of water, it should be, the test tube should be shaken well, so that in order to try to dissolve that sample in that. So after shaking it properly, we need to see the test tube for either of the following two observations. So the first observation may be that the compound is soluble in water. If the compound is soluble in water, it indicates the presence of monosaccharides or disaccharides. One more observation also can be seen, that is, the sample may not dissolve in the water. That is, the sample is insoluble in water. If you get such observation, it gives a inference that the carbohydrate sample what you are taken for the analysis is a polysaccharide. Next comes iodine test. So this particular iodine test, here the major reagent we are using is iodine solution only. Okay, so how does this iodine solution is prepared for this test? A 2% solution of potassium iodide is prepared here. The iodine is added slowly until a deep yellow color is produced. Iodine is not very soluble in water. Therefore, the iodine reagent is made by dissolving in water in the presence of potassium iodide. Because of this, it leads to the formation of polyiodides, which are soluble in water. Just keep it in mind. So, when you are preparing the iodine solution, directly you are not adding the iodine to the water. Initially, you are preparing the potassium iodide solution, that is 2% potassium iodide solution is prepared. Then to that, iodine is added slowly until a deep yellow color is produced. Why it is so? Because iodine is not very soluble in water. To make it freely soluble, we need to add it into the potassium iodide solution. By doing so, what is happening? So when you add the iodine to the potassium iodide solution, there is a formation of polyiodides, which are in turn freely soluble in water. So this is with respect to the iodine solution preparation. Now you can see the principle in the form of the reaction, that is potassium iodide plus iodine. When you mix it, you can see there polyiodides formation. And also in the next line you can see the starch, which is usually present as a polysaccharide. No doubt there are different polysaccharides are also present. So when we will be carrying out the polysaccharide test in the labs, we will be taking care of using the starch as one of the sample. So that starch contains two important components, that is amylose and amylopectin. So when you carry out the test, what is going to happen is the amylose, which is having a coiled nature, a spring type nature. So you can see there in the reactant side. So when you are adding the iodine solution to that, the iodine gets trapped inside that particular amylose. So that gives rise to a complex that we are referring as, there is a color formation due to this. So that indicates the sample is a polysaccharide and in turn it is a starch, which is a polysaccharide. So coming in detail with respect to the principle, the amylose component of the starch has a helical structure. It has got a helical structure, that is coil structure. When it is treated with the iodine solution, the iodine gets trapped inside the coil-like structure, as you can see there in the product side. And the complex has an intense blue color. When the amylose solution is subjected for heating after the color formation, when it is subjected for heating, the helical conformation is disrupted, means it gets broken down, that whatever the helical structure is there, it gets disrupted and it loses its capacity to bind with the iodine. Because of its coiled nature or helical structure, it was capable of trapping that iodine. When you are heating, you are supplying an additional heat to that, which is resulting in the disruption of that particular coil structure, whereby the iodine which was trapped will come out due to the disruption of that helical structure. Further, on cooling, the original conformation is regained. When you subject it for cooling. After heating, the color disappears, but when you keep it again for cooling, the side when the test tube is kept aside for cooling purpose, original conformation is regained. That is, the helical structure is reformed, which in turn traps the iodine, whereby the color is regained. Amylopectin and glycogen, because of the branched structure, gives purple or red color with iodine. So you need to also know, I initially explained it with respect to the starch, the principle. As we all know, the glycogen is also a polysaccharide, and similarly amylopectin is again a counterpart of the amylose in the your starch sample. They will also give the color, but they will give the purple or red color because of their branched structure. They will give the purple or red color when they are treated with the iodine. Coming to the procedure. So here, as I initially discussed with you, we are preparing the iodine solution. So that solution is made to react with 2 ml of the test solution. That is 2 ml of the test solution we are taking. To that we are adding the 2 drops of iodine solution. So when we do it in a test tube, there if it is containing the polysaccharide, that is a starch, there is an appearance of the blue color. So further, as I discussed with you in case of the principle, after the formation of the blue color in the test tube, for further confirmation, if you go for boiling that on a Bunsen burner, so when you boil for 2 minutes, there is a disappearance in the color. Whatever the blue color has been formed, the blue color disappears. Once it disappears, if you keep that test tube aside for cooling purpose, again the regaining of that color takes place. That is, the blue color reappears, which is used as a confirmation that initially when the blue color is formed, it indicates the presence of starch. Further on heating and cooling, if the color disappears and reappears, it is a further confirmation that starch is present. That is, it can be considered as a confirmation of starch in the given sample. Also, it has been said that if the amylopectin or the glycogen is given as a sample, because of their branched structure, they too react with the iodine, but they will result in the formation of either purple or red color. So at the top, I have given you the image where I have tried to show you that the blue color, purple color or somewhat reddish color, which are referred for the confirming the color produced in the reaction. Let us move into the next test, that is Benedicts Test. So it is named after the American chemist Stanley Rossiter Benedict. So basically this Benedicts test is done by initially preparing the Benedicts reagent. So there are 2 types of the reagents of the Benedicts, that is Benedicts qualitative reagent and Benedicts quantitative reagent. So Benedicts qualitative reagent is used to just identify the presence of the carbohydrates, whereas the Benedicts quantitative reagent is specially prepared to know the concentration of the carbohydrate present in that. Okay, so that is the difference. So let us see the Benedict solution which is used for the qualitative test. What all it contains? It contains sodium citrate, also contains sodium carbonate and copper sulfate. These are the main three chemicals which we are using to prepare Benedict's reagent. Let us see the principle. Here you can see a reaction where the water plus copper sulfate will undergo the reaction. The copper hydroxide is produced. So when this particular solution, when it is subjected for treating with reducing carbohydrate, the cupric ion will get in the form of cuprous. So thereby there is a specific color formation with respect to the amount of the special carbohydrate present. That is, here we are referring it with the glucose. So what does exactly the principle gives the information? In this test, the copper sulfate hydrolyzes to form cupric hydroxide, which in turn reduced to cuprous oxide on heating with a reducing carbohydrates. So there are the carbohydrates which are reducing in nature, which you are referring as a reducing sugars. So this test is carried out to identify the reducing sugars. In this test, alkaline medium is provided by sodium carbonate and sodium citrate, which are used to keep the precipitation of cupric hydroxide in solution form. So let us see the procedure involved in the Benedict's test. So here we are taking 5 ml of Benedict's reagent plus 8 drops of test solution. Then these two things are mixed well and they are boiled for 2 minutes. After boiling for 2 minutes, they are allowed to cool. Then we will see for production of the color in that particular test tube. So there may be either formation of a green color or yellow color or orange color or red precipitate. Okay, there may be either green or yellow or orange or red precipitate based on the concentration of that reducing sugar present. What inference we can get? It indicates the presence of reducing sugars. And you can see the above image what I have given here. You can see the different colors where the variation of the colors is primarily due to the concentration of the reducing sugars based on how much amount of the reducing sugar is present in the given sample. It is the different color you can observe. As you can see from the above information what shared, so if you get a blue solution at the end of the reaction, it indicates that there is no reducing sugar present in the given sample. If you get a green or yellow PPT, it indicates that there is a presence of reducing sugar, but that is in the trace amount. If you get an orange red PPT, it indicates there is a moderate amount of the reducing sugar present in the sample. If you get a brick red PPT, as you can see as in the last test tube, so it indicates there is a large amount of the reducing sugar present in the sample. So this is what you need to keep it in mind in order to, no doubt, when you are doing the qualitative test, definitely you are just identifying the presence or absence of the reducing sugar, but if you pay concentration and if you do it accurately, even you can get an idea regarding whether the given sample is containing the reducing sugar or not. If it is containing reducing sugar, in what concentration, whether it is in the trace amount or in the moderate amount or it is present in the large amount. Coming to the next test, that is Felling's test. So this test is having a lot of advantages. Let us see what are they. The reagent, the Fellings reagent, what you are preparing is very stable in nature. The first thing what you need to keep it in mind. And the test is very convenient to perform. Then one more advantage is, since sodium carbonate is a very weak base, the destruction of the carbohydrate does not take place when you are using it in the preparation of the solution. That will not cause any problem. Then it is a semi-quantitative test also. This you should keep it in mind. It is a not only a qualitative test, it is a semi-quantitative test. If the solution are taken in a correct proportion and the procedure is followed strictly, the approximate concentration of the carbohydrate can be judged from the color of the precipitate form. That's what I just explained you with respect to that Benedicts. That also holds good with respect to your, that is, Fellings test also. So this particular Felling test is named after Hermann Christian von Felling. In this test, the presence of aldehydes is detected by reduction of copper to a red PPT of insoluble cuprous oxide. This test is commonly used for reducing sugars but not specific for aldehydes. This you should keep it in mind. So here I have just shown you the principle again. The cupric hydroxide will be treated with the aldehyde, where the cuprous oxide is formed along with the liberation of an acid. So here in this test, very important thing we need to see is, here we are not using only one reagent, we are using the two reagents. No doubt, they are both are Fellings only. They are referred separately as a Fellings solution A and Fellings solution B. What they contain? Then if they are two different reagents, then automatically they need to have the different constituents. What are they? The Fellings solution A contains copper sulfate and water, whereas Fellings solution B contains potassium hydroxide and sodium potassium tartrate. This sodium potassium tartrate has also got another name, that is Rochelles salt. So coming to the principle which is involved in the Fellings test, the carbohydrates having a free aldehyde or ketone group possesses reducing property, just like the simple aldehydes or the ketones. The reducing carbohydrates can be detected by several tests based on the reduction of the certain metal ions like copper, iron, bismuth, mercury, etc. The copper sulfate present in Fellings reagent hydrolyzes itself to give cupric hydroxide, and this cupric hydroxide is further reduced to cuprous oxide when it is treated with the reducing carbohydrate. That's what the principle here refers to. The color of the solution changes from blue to red. Initially these Fellings A and Fellings B, they are having the blue color. If there is a presence of the carbohydrate, when they undergo the reaction, they will in turn give rise to a color that is red. The carbohydrate is simultaneously oxidized to the corresponding aldonic acid. For example, glucose to gluconic acid. If the glucose is there, it will get converted to gluconic acid. The reduction occurs best in the alkaline medium. This is very important. How it is provided? It is provided by the presence of the potassium hydroxide in your reagent. The sodium potassium tartrate, whatever you have used in your reagent preparation, it keeps the cupric ions in solution as cupric hydroxide and has a tendency to precipitate it. This particular test is given by all monosaccharides, maltose and lactose. Keep it in mind, this is very important. This test is given by all monosaccharides plus maltose and lactose, which are disaccharides. As I initially said, there are some advantages with respect to Fellings. Automatically there are some disadvantages also. So what are the disadvantages associated with the Fellings test? So the strong alkali potassium hydroxide present in the reagent sometimes can destroy the carbohydrate. Also, one more disadvantage, auto reduction may occur, resulting in the false positive test. There may be auto reduction may occur sometimes, which may give you a false positive test. So because of such false positive test, compared to the Fellings test, we are preferring the Benedicts test more. This you should keep it in mind. Coming to the procedure, what is involved in case of the Fellings test. So in the procedure, we are taking 2 ml each of Fellings A and B solution, means 2 ml of Fellings A and 2 ml of Fellings B solution. To that, we are adding 1 ml of the test solution, and after that we are boiling this mixture for 2 minutes and we are subjecting it for cooling. After doing so, if you observe the reddish brown PPT, it indicates the presence of reducing sugar. And above, just I have shown you. So when you perform this test, based on the observations you are getting, how you can get to know that it is positive test or negative test. The positive in that sense, that is there will be a presence of reducing sugar, or if the negative means it is an indication that there is no reducing sugar present. So as already been discussed, the Fellings A and B solution will be blue in color. So if there is a no reaction, or if there is no presence of the reducing sugar in the carbohydrate, automatically they won't react and there is no formation of a red color. So the solution in the test tube, even after heating and cooling, it remains as a blue, whereas if there is a presence of a reducing sugar, it definitely reacts with the Fellings A and B solution, whereby there is a formation of the red PPT, which in turn indicates the presence of reducing sugar. Coming to the next test, that is Barfoids test. This Barfoids reagent is devised by Christian Thomson Barfoid, that is in the year 1899. He was a Danish chemist. So this is used to distinguish between the monosaccharides and the disaccharides. No doubt they are reducing sugars identification test, but we need to further differentiate the reducing sugars as monosaccharides and the disaccharides. In that, this Barfoid test is very much useful. So what is the composition of this Barfoids reagent? So it contains copper acetate monohydrate, then glacial acetic acid and water. What is the principle involved in the Barfoids test? Again, you can see there the cupric ions in an acidic medium gets converted to cuprous ion, and which will be getting reacted with the reducing sugar present based on the observation of the color produced, we can get to know that whether the monosaccharide is present or the disaccharide is present in the given sample. So coming in detail with respect to the principle, the aldoses and ketoses can reduce cupric ions even in acidic conditions also. Monosaccharides react very fast, whereas the reaction with reducing disaccharides is slow. This is very important. Monosaccharides react very fast, whereas the disaccharides react very slow. This property is used to distinguish a reducing monosaccharide from a reducing disaccharide. Since acidic medium is unfavorable for reduction, only the strong reducing carbohydrates, that is monosaccharides, give a positive test. So it is mainly for the monosaccharides, and that to identify the reducing sugars, whether they are mono or disaccharides, this test is useful. Coming to the procedure, what is the procedure involved in this? Let us see the reagent, that is we are taking 1 ml of Barfoids reagent and we are treating it with the 1 ml of the test solution, and it is boiled for 2 minutes. So when we do so, we can get two observations. That is, there is a formation of red PPT within 90 seconds, that is within 1.5 minutes. When we are boiling for 2 minutes, within 90 seconds, we need to look into the time. So if you get it within 1.5 minutes, the red PPT, it indicates the presence of monosaccharides. If the red PPT is formed on prolonged heating, that is more than 2 to 3 minutes, if the red PPT is formed, that is on prolonged heating, it indicates the disaccharides, because already in the principle we got to know that the monosaccharides react very fast, whereas disaccharides react slow. That is, they will reduce the cupric ions in acidic condition slowly, the disaccharides, whereas monosaccharides will react and reduce very fast. You can see the color formation in the above image what I have shown, so which can give you an idea with respect to the formation of the color. First we will be checking whether it is containing the reducing sugars or not. If the reducing sugars are there, then whether it is a disaccharide or monosaccharide. So first test tube is clear indication that it is a negative test, means your sample is not at all containing such type of the reducing sugar. Whereas if at all it is containing a reducing sugar, whether it is a mono or disaccharide can be identified by the formation of the red PPT. That depends on the time. If there is a formation of the red PPT within 90 seconds, it is an indication of the presence of monosaccharide in your sample. If the red PPT is formed but it has taken a long time, more than 2 to 3 minutes, it is an indication that there is a presence of disaccharide in your sample. Coming to the next test, that is Salinovs test. So let us see the principle involved in the Salinovs test. So in this we are making use of the Salinovs reagent. What is its composition? It is containing resorcinol, concentrated hydrochloric acid and water. The concentration of HCl is about 12%, as the strength of concentrated HCl is 36. This you should keep it in mind. Whatever the concentration of the HCl we are using is in this reaction for the preparation of the reagent is of 12%, whereas actual strength of the concentrated HCl is 36%. So This test is also known as resorcinol reaction. So that also you need to remember. So when we can see the reaction, we will get few idea, that is with respect to the principle. The carbohydrates are converted into perfurol derivative by concentrated HCl. The perfurol derivative of ketohexose condenses with resorcinol to form a red complex. You can see there. So you have to see that here we have just mentioned that H2SO4, it should be concentrated HCl. So when the aldose or ketoses are treated with the concentrated HCl, dehydration followed by perfurol derivative formation, which in turn on reaction with the resorcinol will yield the cherry red colored complex. When the cherry red colored complex is formed, it is understood that our test is containing basically the carbohydrate which is either a ketose or a aldose, to identify whether our carbohydrate, what we are identifying, whether it is a ketose in nature or a aldose in nature. This test is useful. Let us see the procedure, what we are following in this test. We are taking 1 ml of Salinovs reagent and we are adding 1 ml of the test solution and we will boil for 30 seconds. The test tube is heated for 30 seconds. There are two observations. If there is a cherry red color is formed quickly, it indicates the presence of ketose as a functional group in that particular carbohydrate. If the formation of red PPT occurs on prolonged heating, it in turn indicates the presence of aldehyde as the functional group in that carbohydrate. So that is aldose is present. If there is a cherry red color formation quickly, it indicates the presence of ketoses. If the red PPT is formed on prolonged heating, it indicates the presence of aldoses. So here I have just shown the color formed in that particular reaction for your clear-cut understanding. Coming to the last test, that is what we are referring as a phenylhydrazine test. It is also known as Osazone test. The two names for the same test are phenylhydrazine test or Osazone test. This is the test for reducing carbohydrates. It is the test for reducing carbohydrates. So in this test, whatever the chemicals we are using, what is the composition of all the other things, let us see. So the major mixture here we are referring as a Osazone mixture. This is prepared by mixing 1 part by weight of phenylhydrazine hydrochloride and 2 parts by weight of sodium acetate plus glacial acetic acid is added. So coming to the principle, when reducing carbohydrates are treated with phenylhydrazine hydrochloride, as you could see there in the reaction at 100 degrees centigrade, maintaining a pH around 4.3, a series of reactions will take place. As you could see there is a formation of series of reactions resulting in the formation of Osazone of the respective carbohydrates. As you can see, the glucose, when it undergoes a reaction with the phenylhydrazine, there is a formation of glucohydrazone. Further, when you are adding the phenylhydrazine to that is additional amount of the phenylhydrazine, it results in the formation of phenylhydrazone, which further on continuous addition of the phenylhydrazine results in the formation of the Osazone, that is the crystal structures which are specific for a specific type of the reducing carbohydrates. When carbohydrate is warmed with excess of phenyl hydrazine hydrochloride, because you can see there in the 3 steps we have made use of the successively the phenyl hydrazine addition. It forms phenyl hydrazone by condensation with the aldehyde group first. The reaction does not stop at this stage. The adjacent CHOH group is then oxidized by a second molecule of phenyl hydrazine hydrochloride, which itself gets reduced to aniline and ammonia. You can see there. So there is a liberation of, when there is a phenyl hydrazone formation, no, you can see along with that there is a formation of aniline and ammonia, which are coming out as a byproducts. The resulting carbonyl group further reacts with the third molecule of the phenyl hydrazine hydrochloride to yield the final product, which we are generally referring as a osazone, that is the crystals. So let us see the test procedure, observation and inference. So you can see this phenyl hydrazine test is also referred as an osazone test. Here we are taking the 5 ml of the sample solution, that is the sugar solution what we are taking here, plus 0.3 grams of osazone mixture we are adding. That is how we have prepared it is given in the bracket, that is 1 part by weight of phenyl hydrazine hydrochloride and 2 parts by weight of sodium acetate. That makes us the osazone mixture. Along with this, 5 drops of the glacial acetic acid is added. Then the whole mixture is boiled on a water bath for nearly 30 to 45 minutes. After heating it for such long time, it is further allowed to cool, and then it is subjected for observing the type of the crystals produced under the microscope. Whatever the PPT or whatever the reaction product is formed in that particular test tube, it is taken on a glass slide where a small amount of that particular product is placed and it is covered by a cover slide, and then it is observed under the microscope for identifying the different type of the crystals, because based on the different shaped crystals what we can see, we can get to know that what type of reducing carbohydrate is present, means which reducing carbohydrate is present. So after doing this, there may be different observations as per the type of the crystals formed. If yellowish needle shaped crystals, as you can see in the diagram here, then it is an indication that the given carbohydrate is a glucose or fructose, because initially we will be getting to know that is a glucose or fructose, because we will be carrying out to identify in the previous test, that is whether the aldose is present or ketose. Based on that we can clearly tell that if the aldose is present, there we can tell that here glucose is present, because again you can see there yellowish needle shaped crystals whatever are formed, no, it is same for both, even though there is a glucose or a fructose. So how you can exactly tell? If the needle shaped crystals are obtained, and in the previous case as you saw that there is a formation of that is where you identified the sample as an aldose containing one or the ketose containing one. Based on that, here you can clearly tell that glucose or fructose is present. Then the next observation what you may be seen is yellowish sunflower shaped crystals. When you observe it under microscope, the osazone crystals, if they look like a yellowish sunflower shaped crystals, it indicates the presence of maltose, which is a disaccharide. Then one more observation can be felt, that is if yellowish ball pricked with needle shaped crystals are seen, that is a ball which is pricked with the needles, if such type of the structure is seen which is yellowish in nature. If such crystals are seen, it indicates the presence of lactose. So when you observe after carrying out the ultrasound test, when you observe under the microscope, based on the type of the crystals formed, that is whether the yellowish needle shaped crystals are formed or yellowish sunflower shaped crystals are formed or yellowish ball pricked with needle shaped crystals are formed, you can differentiate them into glucose or fructose or maltose or lactose respectively. Finally, here I just wish to show you a chart. So this is the chart what you need to keep thoroughly in your mind when you will be performing the qualitative analysis for identifying the given carbohydrate sample. So when an unknown sample is given to you, first the test what you will be doing is Maulish test. Based on that, you will get to know that whether carbohydrate is present or not. If you get a positive test, it indicates the carbohydrate is present. If you get negative, it is an indication that the sample doesn't contain carbohydrates. So there is no fun in continuing the carbohydrate test. You can stop there itself. Suppose if you get that carbohydrate is present as a positive result from the Maulish test, further you will go for doing the solubility test, whereby solubility test you will get to know that monosaccharide, disaccharides or polysaccharides are which one is present in your sample. Then you will do the iodine test. If the iodine test is positive, it indicates the polysaccharide is present, like example starch or glycogen, like that. If you get a negative test, it is an indication that mono or disaccharide may be present, whereas polysaccharides are absent in other terms. Then, if the result is negative, you will proceed further. If you get a positive result there itself, the iodine test is an indication that starch is present and you will confirm also, so you will wind up there itself. If you get a negative test of the iodine, then it is an indication that mono or disaccharide is present. Then you should try to further test to identify which is that mono or disaccharide present. So first you will be carrying out Benedicts test, which we are referring as a Felling's test also. That is Benedicts and Felling's test. Both we will be carrying out to identify whether the reducing sugars are present or not. So at a time we can take, because we need to carry out both the tests. No doubt Benedict's test is much effective compared to the Felling's test, because auto reduction what we have seen in the principal study. So the results with respect to Benedict's test are more valuable, but also we will be carrying out the Felling's test also. So when you carry out Benedict's test as well as Felling's test, if you get a positive result, it is referred as reducing sugars, that is mono or disaccharide are present. If they give the negative result, it is an indication that non-reducing sugar is present. Non-reducing sugar is sucrose. No doubt it is also a disaccharide, but it is a non-reducing sugar, sucrose. Then if you get a positive result from the Benedict's and the Felling's test, you should move ahead with the Barfoids test. So you can see there, when you carry out the Barfoid test, again you can expect two results, two observations, positive or negative. If you get a positive result, it is an indication that monosaccharide is present. If you get a negative result, it is an indication that disaccharide is present. Next, if you get the positive result from the Barfoids test, it encourages you to further go for identifying the actual carbohydrate. So for that we will be doing the Salinovs test. So when you carry out the Salinovs test, again you can expect two type of the observations. One is positive result, another is negative result. If you get positive result from the Salinovs test, it indicates the presence of keto sugar, that is fructose. And if you get a negative result, it indicates the presence of aldoses, that is glucose. So further we will be carrying out the Osazone test for both. Whether the fructose is present or the glucose is present, we are going to get the needle type crystals. But based on our Salinovs test, we will get to know that whether the needle shaped crystals obtained from the Osazone test are of fructose or of the glucose. So thereby we can give the final confirmation, that is final report. Now coming to the negative test of the Barfoids. So if you get a negative result from the Barfoid test, it indicates the presence of the disaccharide, and that disaccharide we are further subjecting it for the Osazone test, and we can see two different crystals formed from the Osazone test based on the type of the carbohydrate present in the sample. If suppose maltose is present, then you can see the sunflower shaped crystals, that is yellowish sunflower shaped crystals. If they are seen, that is an indication that maltose is present as a carbohydrate. And suppose if you see the yellowish crystals which are looking like a ball pricked with a needle, then you can see that the lactose is present as a disaccharide in the given sample of carbohydrate. So this is the thing what I wanted to make you aware with respect to the concept of qualitative tests associated with the carbohydrates. I hope now you are better knowing the concepts associated with the qualitative tests, where I just try to bring you to the notice of the importance of the qualitative tests, each qualitative test, where I gave you in brief, that is the principle associated, the composition of the different reagents which are used in that specific test, followed by the procedure followed, observation and inference, what we are getting from that particular test performing. Then also have tried to give you or make you feel that type of the color produced in the test tube when you carry out that particular test. So that's it, what I wanted to give you the information with respect to. Thank you.